Hi, good morning, everyone. Thanks for introduction. I'm from Japan, and I'm nice to meet you, and also nice to meet you again. I'm quite appreciated for the organizer of committee to give me this opportunity to talk something here. 2 years ago, I attended this first conference in Khon, and quite impressive at that time. This time, due to COVID-19, we cannot meet together like last time. But thanks for advantage communication technology, so we can do like this online conference. It's great. Today, what I'm going to talk is about EV, not electricity car, is extracellular vesicle. Mainly, I'm going to talk on role of parasite-secreted extracellular vesicle in immune evasion and parasitism. My talk will include these 3 parts. One is on EV, and the second on Parasite EV, and 3, what we have done on Chichenera EV. Okay, I believe you know well on EV. Here, just give a simple introduction. What is EV? In fact, as early as in 1960s, about 60 years ago, researchers has found this kind of vesicle or small particle along the culture cell. But until 2011, about 10 years ago, this kind of particle was named as extracellular vesicle.
Excuse me, Professor.
Yes.
Now your presentation is in the presenter view. So could you please make your presentation bigger in the big screen?
Oh,
sorry. Yeah.
I had this kind of problem.
Yes. Now the presentation is a little small and
bigger. How can I do like this? Let me see. And wait a moment.
So I think you can switch screen to the second screen for sharing.
Okay. Wait a moment. I think.
Or you can do the presentation as usual, and I will tell you how to switch.
Wait a moment. Let me close up this first. What problem? I don't know exactly, but so I will open my PowerPoint first, okay? And, then I will give the share screen and this one. And then I will give the slideshow. Is this okay? No?
Please wait a sec. It still didn't display.
It's small, you mean? How about this one? But
okay. Thank you very much. It's already full screen.
It's full screen.
Yes, thank you very much.
Wait a moment. I need another way. In this case, it's not full screen. Okay, it's full screen or not?
This one is not full screen.
Okay. I'm not sure why this happened. Okay, this is full screen.
Yes, this is full screen.
Okay. I'm sorry. I'm not so familiar with this Zoom because in our university we usually use the team. Okay. Anyway, I'm sorry for this one. And then I have introduced about this one. This is full screen and still cannot show. Really, is this full screen?
Yes, now everything is okay,
It's okay.
Professor.
Okay.
Yes, thank
Okay.
you so much. Thank
Mhm.
you.
So I just repeat some, but in my screen is not so good because many things interfere my vision. And, how can I do? I'm sorry. Yes, I should. Okay, I will continue now. And first, since I want to give a simple introduction about this EV, extracellular vesicle, and in fact, as early as in 1960, researcher has found this kind of smaller particle around the cultured cell. But until 10 years ago, this kind of particle was named as extracellular vesicle. And there are many now. There are two kinds of vesicle was named as EV. One is microvesicle, a little bigger size, about 50 to 1,000 nanogram. And another is exosome, the size a little small, about 50 to 150 nanometer, nanogram, okay. And the characterization of the EV, just like this, this is a very small particle released from nearly all kinds of cells, from bacteria to mammal cell to human cell and to plants, of course, include from parasites. And another is the vesicle with the membranous structure, the same membrane as cells. And within the EV, there are many substances, and such as protein and messenger RNA and microRNA. This is one kind of short RNA. And also there are many markers on the surface of the vesicle, and there's repeat too. As to function, in the early studies, researchers think this is one kind of way for cell eliminate its waste unneeded components. But now we know this kind of EV has many functions. And if simple to say, the main function is a communicator between cells. What is origin of the EV? There's, as I said, there's two kinds of particle was named as EV. One is exosome and one is microvesicle. The origin is different. Exosome original from in the cytoplasm from the early endosome by this inward body of a membrane. Then it will lead to the formation of MVE, multivesicle endosome. With it contain several exosomes. This kind of exosome is released outside the cell by membrane fusion. Okay. On the other hand, this microvesicle is origin from the cell membrane by outward body to form this kind of EV released like this. In fact, it's difficult to know the origin of the EV in your correct sample because by using the general method, as you know, usually we collect the EV by ultracentrifugation. But there are many characterization for collected EV. One is this composition of EV is quite complex, quite diversity depending on cell types. That means different cells that contain different EV. And also this EV composition of EV is cell type is specific, it's similar to the last one. And also this EV composition is influenced by the physiological or pathological status. So that means this one can be used, that means the EV can be used as biomarkers for some disease such as cancer or for parasitic disease. And however, there are some marker, there are some states shared by EV, even the EV from human or from plants or from parasites. They share similar kind of substances. So we name this as EV markers, such as these 1433 and Alicus and HSP70 and 90. There are many here, showed here. So usually we identify our collected EV, whether this is EV or not. We use this kind of marker to confirm it. Okay, next, how to. Here I cannot see my title because of. Okay, next is how to EV to play its role. We say that EV as a communicator between cells, how they do. So one is the donor cell, they secrete EV like this kind of small particle. There is with the membrane, cell membrane structure. The main way is have this three way. One is membrane fusion, because this is cell membrane. The membrane can fusion with the recipient cell. Then they release the cargo into cell to play its function. Another way is endocytosis. That means the EV is eaten by the recipient cells. And the third way is by the receptor-mediated uptake. And that means on EV, there's some marker. This kind of marker can combine, specifically combine with the receptor on the surface of the recipient cells. Then they can release their cargo substances into cell to play its roles. What a basic role for EV? If we say simply, that's when intercellular communication by exchange protein, nucleic acid, and lipid. This is one simple example to show how EV plays a basic function by changing its substances. This is immune stem cell. This is immune cell. The immune cell was the immune cell can release the EV. This kind of EV can affect the immune stem cell. Immune stem cell. This kind of factor include such as motivate migration and deployment of the stem cell. So this as a response, stem cell will release the EV. This kind of EV will affect the macrophage. So such as inhibit the proliferation or differentiation of activation of the immune cells. On EV, the most studied is on cancer EV. Here, I want to show this kind of experimental how this kind of EV plays a role in cancer progression. There are several ways to communication for cancer, from tumor to stroma, from stroma to tumor or stroma to stroma, these kind of communication ways. Such as from tumor to stroma, tumor cell, they secrete, they release the EV. This kind of EV will affect the stroma cells, such as will play role in angiogenesis or vesicle weakness of the endocellular cells, such as they secrete the EV, have an effect on the macrophage or immune cell to the pre-metastatic IL-6 production, or the EV will affect many kinds of immune cells to play a role in immune suppression. And then, such as this, from stroma to tumor, that means the stroma cell will secrete the EV to affect the tumor cell. For example, this fibroblast, the EV from fibroblast will affect the tumor therapy resistance. Also from the immune cell, they will promote the tumor growth or tumor metastasis or tumor cell evasion. Also, this kind of communication can be seen in tumor to tumor, such as this is aggressive tumor, this is general tumor. From aggressive tumor, you can release the EV to affect the general tumor cell by transfer oncogenic signaling or tumor heterogeneity, or phenocopy the metastatic behavior. So next, what the potential of EV? That means what kind of application for EV can be used for? We know besides the EV plays a role in physiology or in pathology, they also can be used for some other purpose, such as a circulating biomarker. The EV released from let's give an example from the cancer cell. They released the EV. This kind of EV contained specific substances, such as protein or some RNA, especially for microRNA. This kind of EV circulates in the blood circulating. So we can detect this kind of circulating biomarker for diagnosis of cancer. Of course, also it can be used for the parasitic disease diagnosis too. For other application, there is a therapeutic agent, as EV as a therapeutic agent, and as a drug delivery systems too. This is one can be used for the cancer therapy. This is an example for the microRNA as a biomarker for cancer diagnosis. We can see many kinds of microRNA have been identified as a biomarker. They existed in the serum or someone in the urine, this kind of cancer, and prostate cancer, it's the urine. So this kind of mark can be used for diagnosis or prognosis for many various kinds of cancers. For therapy agent, EV can be used to treat some disease This is one example, the EV from the immune stem cell. By inject the mouse, this kind of EV can attenuate ulcerative colitis. This is one kind of autoimmune disease in colon. The mechanism, just like this kind of EV can affect the macrophage by inhibit the M1 macrophage, inhibit the apoptosis, and by promote M2 macrophage polarization. So, and the EV can be used for the cancer treatment. This is one example for this one. We know EV plays important role in cancer progression. So if we remove or block this kind of EV, can be new way to therapy of the cancers. So such as by eliminate of circulating EV using antibody, or by inhibition of EV circulation, don't allow cancer cell circulate EV or stroma cell by using antibody or medicine or some microRNA too, or by disruption of EV absorption to the recipient cells and using such as protease K or some inhibitors. Let's forget this, forgive this one, forget this one. Okay, and now EV, let's say it is still young research field, but it is quite forecast in recent years. We can realize this one from the publisher number. Maybe 10 years ago, very few, but just within 10 years, the publisher number greatly increased. Last year, more than 5,000. And it is saying in Parasite EV, we can see it is increased, greatly increased in the 10 years. And last year, the publication number more than 100, similar in Hermes EV publications. Okay, next I would like to talk something about Parasite EV. And is there Parasite EV? Of course, there is. There have many studies indicated there is EV from parasite. This is a list, some example from the protozoa, EV from protozoa, such as from Leishmania, from trypanosoma, from malaria, or from some other protozoas. And mainly, this kind of EV has a function in modulate immune responses, but also some EV, not from parasite, from the host infected cell here, such as Leishmania from infected cell. The EV from infected cell can affect the immune response or promote parasites by too. Such as some EV from malaria infected cell that can induce the gematogenesis, or that can reduce the cell-to-cell communications. And this is some example for EV study on the nematode, such as from filaria or from some gastrointestinal nematode here and here. And some EV study on trematode. Here there is a liver fluke, Opisthochus, or from the fasciola or from schistosoma. There are many research work, and also from tapeworm, eevee, from the echinococcus too. Okay, how does parasite EV plays a role? This is quite simple, but easy to understanding explanation. Parasite, they secrete the EV for parasite to crosstalk. This also, the parasite EV can affect the host cell. As a response, host cell secrete the host EV. This kind of EV can affect parasite. Yeah, just like this, quite simple. And next, I want to give a little detailed explanation for how parasite EV to play a role. By now, the main target for parasite EV, there's two kinds of targets. One is immune cell, another is cell in direct contact with parasite. Parasite, they secrete the EV. This kind of EV can affect the immune cell, such as macrophage or DC cell or Th lymphocyte. And mainly plays a role in such as modulation of inflammation or proliferation or anti-Helminth response. Also, this kind of EV can affect directly on the host cell, direct contact with the parasite, such as this one is most typical. This is cholangiocyte and the liver cell. This is in liver fluke or some intestinal parasite nematode, such as intestinal nematode. They can release EV to affect this kind of cell directly. Yeah. The role is quite various. There are many roles, such as here list some example, modulation of proliferation and pathogenic effects or wound healing or tumorigenesis. Tumorigenesis mainly on liver fluke. I'm not sure, but it was considered. This is another example to show the role of the Helminth EV on the immunomodulation. So Helminth EV can have effect on the separation of Th1 responses by inhibit the M1 macrophage or neutrophils. So this one, they can inhibit Th1 responses. And also the second, they can, this stimulation of Th1 responses. It looks a little contradictory with the first one, but as we know, there are many kinds of substances in EV. Maybe some substances may be responsible for separation of type 1 Th1 response, or some substances may be responsible for stimulation of Th1 response. So this kind of stimulation also through the macrophage by promote M1 macrophage polarization or through the DC cell too. Of course, this the second role is related to the immunity, especially related to the protective immunity. The third one is induction of the Th2 heterogenetic phenotype. This kind of role is related to the parasite survival and also related to the anti-inflammatory, such as many research show parasite infection can inhibit autoimmune disease or allergic responses. And their function by the promote M2, macrophage polarization, or affect the TDC cell. So this one, they can promote the T-reg response. Also, this kind of function can inhibit the eosinophil response or ILC2 response too. The fourth is stimulation of Th2 response. Someone like this, like the first one, second one, and they can initiate the Th2 responses through the promoted Th2 cells. And this kind of response related to the parasite expulsion. Yeah. This slide shows the role of the EV, parasite EV, in protective immunity. They have some studies to show that, such as in this gastrointestinal nematode, by vaccination with the EV, they can decrease water burden by 82%, such as this Chichenera's vaccination can decrease the water burden by 60%. So this indicated there are some substances within EV can be as a candidate antigen for vaccine for protective immunity. So maybe this is one new method, new way to identify some good candidate for vaccine development. Here, I tried to summarize the role of the parasite EV. One is immunomodulation and immune evasion such as in the parasite Leishmania or Echinococcus filaria or gastrointestinal nematode. They plays function mainly by inhibiting the macrophage and the DC cell. The second is protective immunity, as I showed in last slide. There have been many reports by vaccination with EV, they can get the water burden, decrease the warm burden or others, PEG. The third one is direct effects on the host cell or infected host cell derived EV on parasite. I have talked this one about such as induced genotogenesis in malaria. That means the EV from the infected cell. Also, EV from the infected cell of the malaria can reduce the cell-to-cell communication. Or in the opistokis, we can see they can promote cholangiocyte proliferation and maybe can promote the tumorogenetic pathway. The fourth one is as a biomarker for diagnosis. This is quite promising, and especially for now, most of it is for microRNA. The microRNA from EV can be used because, as we know, detect antigen from the circulating from serum is a little difficult by now. Many research has shown it's not so easy, but detect the microRNA is possible. So by now, there are many studies have shown high sensitivity and specificity for the schistosoma diagnosis. Also, some antigen from the EV was identified as a candidate antigen for diagnosis from the fasciola or echinococcus. Okay, last part, I would like to talk what we have done on Chichenera EV. In my lab, main work is on Chicheneras. And this is Chichenera spiralis in its life cycle. There are two developed stages. One is adult worm in intestine, another is muscle larva in muscle cell. And human or host usually infected by eating the infected pork. Within the pork, there is muscle larva. The muscle larva can invade the mucus membranes in the intestine, then develop into adult worm. This kind of adult worm can give rise to newborn larva, very small larva. This kind of newborn larva can invade the blood vessel and then throw the blood stream to the muscle. Then they invade the muscle cell. Within the muscle cell, they can develop into the mature muscle cell, where they can live long time, maybe 10 years or 20 years. So Chichenera is quite an interesting parasite. It's quite unique. It's biggest intracellular parasite. Also, there is several different, quite distinct developed stagement. Adult worm and muscle larva, both they are secreted products. We call these ES products. Until recently, we know within the ES products, there is EV from adult worm and from muscle larva too. In all this study, the main purpose, we want to know the role of Chichenera EV in survival. That means its effect on the immune response, and the role of adult worm EV in intestinal parasitism. We just speculated the role of adult worm EV and muscle larva EV is different because the parasitism is different for both stages. The third one is the role of muscle larva EV in muscle cell parasitism. And the final purpose is we want to see the difference between adult worm EV and muscle larva EV. This is the result, first result. TSAD EV, and this means that the EV from Chichenera spiralis adult worm, and TSML EV, that means the EV from the muscle larva. We co-cultured this kind of EV with the macrophage, then activate the macrophage, use LPS, then measure the R6 in the culture media. So from here, we can see both the EV from muscle larva or from adult worm has can inhibit, greatly inhibit the activation of macrophage. So there's nothing here. So the IL-6 expression becomes lower. Also, this is gene expression. Okay, this is the protein concentration within the culture medium. We can see the EV from both adult worm and muscle larva can inhibit the macrophage activation in a dose-dependent manner. This is another evidence to show the EV can inhibit macrophage activation by the flow-cytometry analysis. We can see the CD80 marker. This is M1 macrophage mark. The percentage was greatly decreased in both EV treated macrophage. Then second one, we want to see the EV effect on the mucus production. As we know, parasite infection in intestine they induce the Th2, induce a Th2 response. This Th2 response can cause the goblet cell become hyperpressure or become mucus hypersecretion. So this kind of mucus can entrap the worm, so make the worm expulsion. So this mechanism we can consider as protective immunity. And here we did an experiment like this, both EV from adult worm and from the muscle larva. We co-culture with the goblet cell line, then measure the mucus production. Mainly these two, we did these two kind of mucus production. So from the result, we can see EV from adult worm here and here, not from muscle larva can inhibit the mucus production. That's a little interesting result. So muscle, EV from muscle larva, they have no any affection on the goblet cell for goblet cell to produce the mucus, but the EV from adult worm have this kind of roles. Then we observed the EV from Chichenera, their role in the nut cell formation. Nut cell formation is interesting and focused topic in Chichenera research. This is also my topic too. I have spent more than 25 years to research this topic. We know the newborn larva invade muscle cell. They give the muscle cell damage and make the inflammation around the muscle cell. So muscle cell, as a muscle cell, host cell, they try to regeneration. We want to regenerate the damaged muscle cell into new muscle cell. But Chichenera don't allow this happen. They want home to live for a long time, for 10 years, for 20 years. So they try to deform the infected muscle cell, try to construct the home by themselves through some mechanism. And finally, just for 2 or 3 weeks, they can change the muscle cell, infected muscle cell, into a completely different phenotype cells. We call this one nut cell. During nut cell formation, there are many things happened, such as activation and proliferation and the transformation of satellite cell. This is one kind of stem cell muscle. And this one, lead differentiation of infected muscle cell. As we know, muscle cell is terminal cell. This is, the muscle cell is a differentiated cell. So Chichenera make a differentiated cell to differentiate again. This is one interest in step during the muscle, during the nut cell formation. So here we want to see EV from Chichenera. Is there EV from Chichenera have effect on the differentiation of muscle cell or not? This is the result. We very simple. We just co-culture with the EV from adult worm and from muscle larva with C2C12 cell line. This is well-known muscle cell line. And then we measure the cell differentiation related gene expression. This one too. This is myogenic and MEF2C. This is muscle cell differentiation markers. So we can see the result here. This is from the muscle. This is EV from muscle larva. Here from muscle larva. This is EV from adult worm, muscle larva, adult worm, muscle larva, adult worm. So we can see the EV from muscle larva, not from adult worm, can promote the muscle cell differentiation. So at least this suggests the EV from muscle larva involved the muscle nut cell formation by secrete its EV. Okay, final. We know there is a different role of EV from adult worm and from muscle larva. So next we want to see the compounds within the EV is different or not. We did the mass analysis. So from the adult worm EV, we detected 240 kinds of protein, and from the muscle larva EV detected 296 kinds of proteins. But only about 27% proteins are shared by both EV, and nearly 30% of protein is specific to adult EV, and nearly 43% is specific to muscle larva EV. So this suggests this kind of specific protein may be responsible for their different or distinct parasitism. Here we just list up some top 20 high expression protein in adult, in the EV from adult worm. Here the expression level in adult worm, but in the EV from the muscle larva, 0 or quite low. This is for the changes. Within this kind of candidate protein, there are several famous ES protein in Chichenera research, such as this one, DNS2, quite a famous one. This is a list for the top 20 high expression protein in muscle larva EV. Also, you can see here the expression label, the muscle larva EV in adult larva EV, in adult worm EV. Also within it, there are several well-known ES proteins, such as this 53 kilodalton ES protein. I'm doing research on this protein too. And also there's some famous one, serine proteinase. And this is a list of the top 20 high expression protein shared by both EV from adult worm and muscle larva. Here we can see the similar expression level here. Okay, finally, what I want to say is parasite EV is still a young research field. There are still many things needed to do, and many things is waiting for us to do, especially in the immunomodulation or immune evasion or immune protection. This will related to the vaccine development and pathological mechanism or diagnosis. So it's not so late for us to act from now. Thank you for your attention.
Thank you very much, Professor, for very impressive talk. We are now in the Q&A session. If anyone have a question, feel free to ask or raise your hand to let me know that you have questions, or the question in the chat box are also welcome. Professor Laha, please.
Yes, thank you.
Hi, nice to meet you again.
Nice to meet you again. Thank you for a very nice talks. I'm glad to hear that you are working on the EV of the Chichenera. We are also working on the EV of the Orpitocus viverrini.
Yeah, I read your paper too.
Yeah, we found that the EV is very important in stimulate the host host parasitic relationship. For example, we found that OV stimulates cell proliferation and internalize into the cell host and cause several pathology. I would like to know that in the Chichenera, do you notice how the EV that's involved in the nerve cell formation? That would be, I think I expect that the EV would be play a role critically in the nerve cell formation. Are you planning to do that investigation, or do you have the idea on that?
Yeah, thank you for your question. And in fact, by now, many research on parasite EV, they just, everyone just finished to the mass analysis, what compounds with the EV. As I showed our result, there is more than 200 kinds of EV within the 2 kinds of protein within the EV. So next step is quite difficult. We have to identify what kind of protein is responsible for their function, their roles, such as in nerve cell formation. What kind of protein? If we try pick up some well-known one, such as DNS2 by now, many speculation say DNS2 from the in the past time, it is named as the 43 kilodalton protein. They plays a role in that cell formation, but no direct evidence. So we only can pick up some famous, well-known candidate protein. Then by do some recombinant, prepare recombinant, then see its effect on the nerve cell formation. By now, only thing we can do that like this, but it's not easy. Too many protein. We don't know what kind of protein can be used as a candidate. That's a problem for me now.
Yeah, that may have a second question, because that point is go to my second question. That I'm a little bit surprised that from your proteomics data, you couldn't find a major protein that is a cargo protein, for example, CD63, CD9, in clicking like that, which is, in trematode, they play function in that sort of thing about the pathology.
Yeah.
Do you know why they don't have that, or there's no that source of protein in the proteomics?
Yeah, I list up just 20 protein. In fact, there is this 20 protein is the highest expressed. But in fact, within our list, 4 list, there is many protein is from the EV marker, such as HSP70 or some other many. I cannot remember all. But not every kind of marker can be detected in my sample. Especially we did not find the EV CD9 or CD63, but at least the other marker, many other marker is detected. So I believe should be okay for our EV.
Thank you.
Thank you.
Thank you, Professor. Any more questions, please? So if you have any questions, you can drop in the chat box also if you're not comfortable to ask the professor directly.
Okay, thank you.
Okay, now we don't have no more questions. So if anyone have the question after the session, you can drop your question and we will send the question directly to the professor and reply you back via the chat box. So thank you very much, Professor.
Okay, thank you.